Organoids are fast becoming one of the most cutting-edge tools of modern life sciences. The idea is to use stem cells to build miniature tissues and organs that accurately resemble and behave like their real counterparts. One can immediately appreciate the value of organoids for both research and medicine. From basic biological research to drug development and testing, organoids could complement animal testing by providing healthy or diseased human tissues, expediting the lengthy journey from lab to clinical trial. Beyond that, there is already the whisper of organoid technology perhaps being used for replacing damaged tissues or even organs in the future. Take stem cells from the patient and grow them into a new liver, heart, kidney, or lung. So far, established methods of making organoids come with considerable drawbacks. Stem cells develop uncontrollably into circular and closed tissues that have a short lifespan, as well as non-physiological size and shape, all of which result in overall anatomical and/or physiological inconsistency. Now, scientists from the group led by Matthias Lutolf at EPFL's Institute of Bioengineering have found a way to guide stem cells to form an intestinal organoid that looks and functions just like a real tissue. Published in Nature, the method exploits the ability of stem cells to grow and organize themselves along a tube-shaped scaffold that mimics the surface of the native tissue, placed inside a microfluidic chip, a chip with little channels in which small amounts of fluids can be precisely manipulated. The EPFL researchers used a laser to sculpt this gut-shaped scaffold within a hydrogel, a soft mix of cross-linked proteins found in the gut's extracellular matrix supporting the cells in the native tissue. Aside from being the substrate on which the stem cells could grow, the hydrogel thus also provides the form or geometry that would build the final intestinal tissue. Once seeded in the gut-like scaffold, within hours, the stem cells spread across the scaffold, forming a continuous layer of cells with its characteristic crypt structures and villus-like domains. Then came the surprise. The scientists found that the stem cells just knew how to arrange themselves in order to form a functional tiny gut. It looks like the geometry of the hydrogel scaffold, with its crypt-shaped cavities, directly influences the behavior of the stem cells so that they are maintained in the cavities and differentiate in the areas outside, just like in the native tissue, says Lutolf. The stem cells didn't just adopt to the shape of the scaffold, they produced all the key differentiated cell types found in the real gut, with some rare and specialized cell types normally not found in organoids. Intestinal tissues are known for the highest cell turnover rates in the body, resulting in a massive amount of shared dead cells accumulating in the lumen of the classical organoids that grow as closed spheres and require weekly breaking down into small fragments to maintain them in culture. The introduction of a microfluidic system allowed us to efficiently perfuse these mini-guts and establish a long-lived homeostatic organoid system in which cell birth and death are balanced, says Mike Nikolaev, the first author of the paper. The researchers demonstrate that these miniature intestines share many functional features with their in vivo counterparts. For example, they can regenerate after massive tissue damage and they can be used to model inflammatory processes or host microbe interactions in a way not previously possible with any other tissue model grown in the laboratory. In addition, this approach is broadly applicable for the growth of miniature tissues from stem cells derived from other organs such as the lung, liver or pancreas, and from biopsies of human patients. Our work shows that tissue engineering can be used to control organoid development and build next-gen organoids with high physiological relevance, opening up exciting perspectives for disease modeling, drug discovery, diagnostics and regenerative medicine, says Lutolf. Story source: materials provided by École Polytechnique Fédérale de Lausanne. Original written by Nick Papageorgiou. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.